15 years ago, we published Our Stolen Future. It was a bestseller, translated into 15 languages. It explored how, in essence, the core of it was looking at how exposure in the womb to contaminants that behave like hormones alter development and then lead subsequently to diseases later in life. What it did was to stimulate, it helped with others involved in the process, but it helped stimulate literally hundreds of millions of dollars of research by governments around the world into asking some of the questions that we and other scientists who were at the time asking. Now we know something is happening. What is happening? Our book was full of questions. It acknowledged the uncertainty. But in the outpouring of results, and tens of thousands of articles have been written about endocrine disruption in the peer-reviewed literature since we published this book, as a result of the investigations, we really know a lot more. There still are questions. But what I thought I would do today would be to rise up to 30,000 feet above the scientific plane and tell you about some core themes that have emerged out of that scientific literature. We're actually living in the midst of a scientific revolution based on these findings, and they're changing how we think about the relationships between contamination and health, particularly, and it's perhaps surprisingly because the initial focus was on fetal exposures, but how those fetal exposures contribute to chronic diseases of adulthood. Some of the discoveries in the science have been completely unexpected, including signaling that some of the materials we have embraced and included within our households, some that we thought were safe actually are not. For me, the most exciting thing is out of this science have come many opportunities for prevention of disease. I'm going to walk you through what I would regard as the 6 central themes of this revolution in science. The first one is that, and this is the most profound, contaminants interact with genes in ways that not only don't reflect current health standards, but also in ways that the traditional scientific tools used to assess toxicity didn't acknowledge. Second, these interactions can take place at extremely low levels. Can you all hear me when I just realized I was shifting away from the mic? Okay, great. Third, exposures in the womb, and this will be a central theme, exposures in the womb can set in motion things that play out over a lifetime. Fourth, these exposures are ubiquitous. Everyone experiences them, and they take place in mixtures that can interact in unexpected ways. Fifth, the tools that we have used in toxicology to assess risk are blind to some of these impacts, and especially several that I will focus on. And sixth, there is now emerging a science called green chemistry that's using this science to guide new chemical syntheses to create a wave of new materials that offer opportunities for innovation, both material innovation and economic innovation, based on materials that are inherently safe. So those are the core themes. I'm going to use this one picture to illustrate 3 of them. How many of you have seen this picture before? Okay. It's from Retha Newbold's work at the National Institute of Environmental Health Sciences. It's the same strain of mouse. They monitored caloric intake over the lifetime of the mouse up to this age, and they monitored the activities level, and they were basically not statistically distinguishable. Same activity, same caloric intake, but obviously 2 very different mice. What's the difference? The fat mouse was exposed to 1 part per billion of a potent endocrine disrupting chemical right at birth. We now know a lot about the science that makes this happen. What's in essence happening is DES and other endocrine disruptors can increase the rate of conversion from stem cells to fat cells, so the mouse grows up with a lot more fat cells than it normally would have, and those absorb more lipids, more fat. The second part of this message, of this lesson from these mice, is that if the animal had been exposed to 1,000 parts per billion, 1,000 times what it takes to make a mouse fat, it would have been scrawnier than the control. So a high-dose experiment doesn't predict what happens at low doses. That's vital when you're considering endocrine disrupting compounds. And third, back to that theme of developmental exposure, developmental exposure led to obesity in adulthood, which gradually developed over the life of the animal. So the 3 key issues there: low levels matter, High-dose testing don't predict low-dose results. And third, early life exposures can impair adult health. So another key element of this revolution, which I have touched on briefly, is we used to think that only high levels mattered because you need a high level to mutate something. You need a high level to kill something overtly. But when you're dealing with changes in gene expression, things happen at low levels. Levels that people commonly experience, levels that couldn't even be measured 20 years ago. If you look at the animal studies, there are links to animal variations, animal versions of each of these endpoints. We don't have epidemiology yet for many of them, for most of them. We don't have human confirmation. There are scientists actively working today on every one of those links. Now, this looks like a very complicated slide. Actually, conceptually, it's very simple. What it shows, it takes data from lots of different animal experiments, and it shows you, in terms of hours after exposure, what are the levels that are in the blood of the animals. In each of these experiments, there were adverse effects on the animals, okay? And so the black line is the average, and it looks like the serum levels are pretty low, right? 0.1 part per billion. Well, the crucial question is, well, how does this relate to what's in people? Are the animal experiments, usually we're told that, oh, those rat experiments use doses that are completely irrelevant to people. So how do these levels compare to the average in people? That's the average level in people in the US. So these animals are being exposed to levels that are lower than those in people, and they're causing adverse effects. There's not a safety factor built in here. The tools that we have used to assess risk are blind to some of the things I've just summarized for you. And I want to ask, well, how is it that regulatory toxicology missed this? And what I would say is that as good a job as they're trying to do, the process of regulatory toxicology depends upon standardized assays, which often are decades old. And you can't deal with some of the phenomena I've summarized today, the cutting edge academic work is focusing on, with old tools. It's sort of like, so imagine, how many of you have seen pictures of the Hubble Space Telescope, the pictures of galaxies out in space? How many of you have seen those? Okay? So imagine this first time you see one of those. You say, that can't be true. And so you grab your binoculars and you go out in the backyard and say, see, it's not true. That's what's happening here. People are using old assays to try and look at 21st century phenomena. It can't be done. Actually, I don't need those pictures. So that's a key part of it. A second part is that there's some assumptions that are used in risk assessment that, for example, underlie how you calculate what a daily acceptable intake is. One is that there are safe doses, that there is a safe dose beneath which exposure is okay. The second is that the dose-response curve is, if it's not linear, it may be curvilinear, but at least it's not non-monotonic. Now, non-monotonicity is a mathematical concept. It means the shape is like a U or an inverted U. What it actually means is that the sign of the curve changes, excuse me, the slope changes sign as you move along the dose-response curve mathematically. But all of regulatory toxicology assumes there are monotonic dose-response curves, that the inverted U or the U-shaped curves don't occur. And not only that, but that allows them to assume that high-dose testing can predict low-dose results if the curves are monotonic. Those assumptions have just now recently begun to be challenged. But even still, the doses that are predicted to be safe, once you do your regulatory testing and you figure out what's the daily acceptable exposure level, that level isn't tested for safety because it's usually about 1,000-fold beneath the lowest dose that's run. Now I want to show you some results from a lab looking at tamoxifen. How many of you know what tamoxifen is? It's key to treating breast cancer. And the reason why it's used to treat breast cancer is shown in this graph. This shows the response of the growth of a human breast cancer tumor to tamoxifen. So at high levels, tamoxifen really knocks out that breast tumor, right? That's what you want. So let's move down the dose-response curve of tamoxifen. And you can see here that you can establish the no observed adverse effect level. It's the first exposure level that doesn't see a significant difference from the control. Okay, makes sense? You're moving down. So that then allows you to begin to introduce several safety factors. One, because rats aren't little animals. One, because kids are different from adults. And one, because there are differences among people. And that takes you to a safe level of exposure based on that calculation. Does that all make sense? That's a classic example of how you apply safety factors to a toxicological experiment. The problem is, for regulatory toxicology, the problem is that this scientist actually did a full dose response curve. That's what happens with tamoxifen at lower levels. And at the very level where tamoxifen was predicted to be safe is the level at which tamoxifen is most stimulatory to breast tumor growth. Okay? That's a problem. Why is that? Well, it turns out that as scientists now know that as you move up the dose response curve, the suite of genes that are being turned on and off changes. That's a classic result in endocrinology that guarantees that you can't predict from high dose experiments what's happening at low doses, because different genes are being turned on and turned off. And in fact, at this highest dose here, it isn't an epigenetic phenomenon at all. It's overt toxicity. It's killing the tumor. And that doesn't involve changes in gene expression, period. So if you are depending upon assays that are looking at that sort of endpoint, you completely miss the epigenetic phenomena that take place at low doses. So if we then go and calculate, based on these new data, what the safe level of bisphenol A is, you establish another no observed adverse effect level, and there it is. That's what's safe. None. So these curves are really this is standard endocrinology. Suite of genes, turned on and turned off, changes across the dose response curve. Standard endocrinology. You learn it in Endocrinology 101. And so the big challenge has been, as we've recognized that some contaminants behave like hormones, they follow the rules of endocrinology, not standard toxicology. That's a big deal. And that's where the rubber hits the road with endocrine disruptors. So there's some bad news in this message. A widespread exposure to compounds that interfere with gene expression, sometimes at extremely low levels. It happens. Today's epidemiology and toxicology is ill-equipped to deal with these effects. But there's good news, and I really want to leave you with that message. I cut out most of it early on just to follow Jackie's admonitions. But what this means is that, like I said, if diseases are linked to genes, we now know there may be ways to prevent them through eliminating the exposures that hijack gene expression. That's awesome. That's hope. That's something we can do. We are going to have opportunities to prevent some diseases that haven't been perceived before as preventable. What's next? There are many scientific questions left. This is a great time. If you're a scientist, this is a wonderful issue to be in today. But I think we know enough now to start incorporating what we already know in a new generation of precautionary health standards, and also to work with green chemists so the next generation of materials not only provide economic opportunity, but protect health. We can do that. If you look at today's human epidemics, here's a partial list. There are scientific data showing plausible links to endocrine disruptors to virtually every one of those diseases. Now, in the end, we're probably going to learn that endocrine disruption is irrelevant to some of them, because the science is moving along. But some of them, we're going to learn, actually, it is one of the prime drivers. And we need to be using this science to guide policy and to take us into an era in which our kids are healthier. So thank you.